All right. Good evening, everybody. So first and foremost, I'd like to welcome everyone to our online showing of Dr. Toxopeus talk. Apologies in advance for any extra noise in the background. I have three budding young scientists who are going to be joining us, and they may pop their head onto my mic at any given time. So, again, apologies in advance. So, first and foremost, before we actually start this, I'd like to just give a shout out to the NSIS. They've been around promoting science in Nova Scotia since 1862. And as many of you may know, and for those who don't, each year the NSIS hosts free public lectures on the first Monday of each month between October to May to provide a meeting place for those interested in science. And this includes from the very, very young to the very, very old. Right now, we are at the halfway point of our lecture series. And just as a reminder, then we then have another three great talks hosted at Dalhousie as well as one at SMU. In terms of just some quick housekeeping, please note that we have our socials through Instagram and Facebook, as well as the official NSIS website, where there is a host of information about our upcoming series, as well as other information that may be relevant to particularly young budding scientists, including some travel awards and other information relating to those interested in institutional memberships, as well as general memberships to the NSIS. And again, this is for anyone who is interested in science. So, if by some chance the two young budding scientists that I met at the Antigonish Farmers Market who were doing a research topic on ticks and how they impact our enjoyment outside, I would strongly recommend, although I know you guys were just in junior high, to go take a look at the NSIS website and just learn more about the organization. Now, before we get into the actual talk, I think it is important for us to acknowledge that we are all tree people and that we are in Mi'kma'ki, the ancestral unceded territory of the Mi'kmaq people. And it is up to us to understand our impact as well as the impact of colonialism. And that the treaties did not deal with the surrender of lands and resources, but in fact recognize Mi'kma'ki and the rules of which ongoing relationship between these nations. Now, in terms of our talk today, I would like to give a not so quick blurb of Dr. Toxopeus. So, Jantina, as she tells her students to call her, she completed her PhD in biology at Western University in 2018. Prior to joining us, she was a postdoctoral researcher at the University of Colorado for 2 years before we were very lucky and have her join us in 2020. Many of us remember 2020 very well. And Dr. Toxopeus, she was recently actually tenured and promoted to associate professor last year, which was expected as her expertise has made her an amazing teacher, mentor, and supervisor. In her time as St. she has published in top-tier journals, including the American Naturalist, which is one of the world's premier peer-reviewed publications in ecology, evolution, and behavior research. She has received over $1 million in funding and has been involved in the training of more than 2 dozen highly qualified personnel in her lab. As we will soon learn in a moment, Dr. Toxopeus' area of research focuses on cell and molecular biology of how insects tolerate cold and survive freezing, which is important for understanding the impact that climate change will have on some of these pests. And so it makes perfect sense that our talk today from Dr. Toxopeus is entitled Ticked Off by the Cold, How Ticks and Other Critters Deal with Winter. So please join me in welcoming, or virtually, or welcoming Dr. Jantina Toxopeus. And before I jump into that, I just want to give a quick question to those of you. How many of you are ready for, and this is from my daughter, tick tock? Anyways, I will leave Dr. Toxopeus to start things off for us.
Oh, thank you, Derek. Very kind introduction. All right. And I'm going to bring up my slides here. Yeah. Okay. Awesome. And yeah, thank you everyone for joining today. It's great to It's sad to not be in person, but I'm happy that you're here anyway, and excited to talk about some of what we do here at St. FX. And I really appreciate the invitation from the Nova Scotia Institute of Science, and I think it's really great that we have lectures like this across the province, including at St. FX. And yeah, it's a really, really great organization. You do a lot of good work. So, whoop, let's get the slides moving, maybe. Uh, hmm, all right, let's do this. There we go. Okay, so like Derek mentioned, I mostly work on insects, and one of the reasons that I care about insects, and I hope you care about insects too, is that they're super important for a lot of reasons. So here we've got a bird that's chomping on a caterpillar. Um, about a third of the bird species in North America rely on insects as food. So if you care about birds, you should care about bugs. We of course know that many insects are pollinators, both the bee that's shown here, but lots of other types of insects too, like flies and beetles. So they're important for like the general ecosystem out there. And there's more insect species than kind of any other group of animals. They're really, really abundant and diverse, and help make the world go around. They're also sometimes a little bit of a problem for us. So there are insects that we would call pests because they interfere with human activities, usually economic activities, although sometimes others. So this apple maggot fly here is native to this region, but they can cause damage to apple fruits and make them harder to sell and export. This cabbage white butterfly is the bane of many gardeners who are trying to grow cabbages or any of their relatives like kale or broccoli. The caterpillars just will just eat all of it. Um, Hemlock woolly adelgid is making a bit of a splash in Nova Scotia at the moment. This is a hemlock, part of a hemlock tree, and these white fuzzy bits on the underside are from this insect called Hemlock woolly adelgid that's invasive, and it's um, tearing up southern Nova Scotia. It's not here yet, but it's probably coming. And that's not great for the hemlock trees. It can eventually cause them to die and we lose some of our old hemlock, old growth forests here. So sometimes, we're not so happy about the insects in our life. And then this mosquito on the right is a reminder that insects can also directly affect our health, right? So some of them are vectors for diseases, and there's some great work on mosquitoes out of Acadia University, actually, trying to understand all the there's many species of mosquitoes, only some of which are a problem for human health, because not because they are toxic, but they carry pathogens that can be dangerous to us. So whether you care about insects because you care about the general function of the planet, or you might care about insects because of what they can do to us, there's lots of reasons to care about bugs. And one more reason is that uh, insects, unlike us, the insect's body temperature will match their environment, right? So they are much more susceptible or vulnerable to changes in environmental temperature than we personally are. So we know that the climate is getting warmer. Um, Winter is changing particularly fast. So not only are summers getting warmer, but so are winters. And this map here shows a projection of sort of what the mid-winter average temperature is, where they're going over the next 100 years or so. And the darker the red, the sort of more dramatic the color change, more dramatic the temperature change is. So especially in the north, there's some really big changes happening in winter. And you might think, well, warmer winters sounds like a great idea. That would be wonderful. Maybe we'd have less snow to shovel. But that can be a problem. If it's warmer and there is less snow, that can make the environment colder for a lot of critters because organisms that overwinter under the snow are actually kind of insulated by the snow layer, so it's warmer under the snow than it is in the general air. So less snow can actually expose overwintering critters to lower temperatures. Um, in addition, winter's temperatures are becoming more variable, so there's more freeze thaw events. And so, it was -20 last week and it's -2 today. And if it frequently sort of warms up above zero and cools down below zero, then that freeze thaw can be quite dangerous for a lot of organisms as well. So I care about insects and I also care about winter and understanding how these changes in temperature, so colder exposure to sort of more extreme cold, either due to less snow or these sort of cycles of increasing and decreasing temperature. I want to understand how that's going to affect the bugs around us and us ultimately. Okay. Okay. So, in my research group at St. FX, we work on a few different, well, we'll work on lots of types of bugs. I'm a bit of a generalist, but our main sort of bread and butter bugs are these critters that you see here. So on the top left, we've got the Springfield cricket, and they're found through a lot of southern Canada and not officially in Nova Scotia, but I have a colleague in the valley who has heard these guys. They're called Springfield crickets because the adults sing in the spring. So they overwinter as this juvenile stage, which is just before they become adults. And then when it warms up in the spring, they complete their life cycle, become adults, and they sing to find a mate. Um, and these insects are really cool because they can survive freezing. So ice forms inside their body, and that would kill a lot of organisms, but not these guys. So they can freeze and then warm up, or thaw back out and be fine. So our lab studies them to try and understand, well, how do they do that? Because if we understand that, we can generally understand perhaps how like freeze thaw events are going to affect insects, but also, it'd be really cool uh, from a biomedical perspective, it would be quite useful to be able to freeze um, human organs and things like that. And right now we can successfully freeze single cells like eggs and sperm, but if you want to try and like preserve a heart, so you can get a heart from a donor and freeze it and then use it when you need it, we're not at that level, right? So maybe we can learn something about these whole animals that survive freezing. Um, on the bottom here, we've got a spruce budworm. This is it's a caterpillar. The adult is a moth. And they are a big pest of spruce and fir trees. They are native to Canada, so they're not invasive like the hemlock woolly adelgid. And every sort of 40 to 50 years, their population explodes and they have what's called an outbreak, and they just kind of devastate the spruce and fir trees in the area. So there's a big outbreak going on in Quebec right now. New Brunswick has been able to kind of suppress it so far, but Newfoundland has not been so lucky. And the outbreak is just starting in Cape Breton here. So I work with these guys because they're important to humans, like they can have a big effect on the timber industry, but they're also just really cool. So they overwinter as these super tiny caterpillars, like a couple of millimeters long. You think about like the millimeter division on your ruler, and like they're just absolutely tiny, and they can survive winter in this tiny state, without having ever eaten anything. And they don't survive freezing, but they can prevent their body from freezing at really low temperatures. So even at temperatures below -20, they will remain unfrozen, which is part of how they survive. So they're a fun group to study as well. And then up on the right is our tick. Ticks are not insects, as they've got too many legs. You can count 8 legs, so you know they're not an insect. But they're related. They have the same characteristics of, their body temperature will match the environment. And um, there's a lot we don't know about how ticks interact with winter. So that's where we're going for the rest of this talk. Um, and I know sometimes I love hiking, and I do hike out here. I just engage in all of the tick safety stuff, like colored pants and tick checks afterwards and that kind of thing. But it does sometimes feel like this is what it's like going out for a hike in the spring or summer. Um, so I do encourage you to not let the ticks get you too down about uh, your outdoor activities. As long as you engage safely, it's we should enjoy our beautiful province. But okay, so why ticks? I mean, I suppose many of you are at this talk because you care about them in some respects, right? And the tick species I study, my lab studies, is the one that can transmit Lyme disease, which is a really weird and complicated syndrome. The symptoms can look very different in different people. This image here just shows sort of some of the things that can happen, and some people have no symptoms, and some people it like ruins their life. So it's a bit of a strange one, but um, uh, the sort of classic, usually people will go get checked out for Lyme disease if they know they've been bitten by a tick and they get kind of a rash. It doesn't always have to have this like bullseye um, shape. And the fancy scientific word for it is erythema migrans. But fortunately, in Nova Scotia, pharmacists can prescribe antibiotics for this. And if you do get bitten by a tick and you're worried about it, keep the tick, even though they're probably grossing you out, because then that will help the pharmacist figure out what to do with you. Um, okay. Or if you happen to have my number, you can text me a picture of the tick and I'll tell you if it's likely to be a problem. I'm no expert on Lyme disease, but my tick identification skills are okay. Um, uh, Lyme disease is particularly prevalent out here. So this is some data from 2020. And this map here, the green colors show, well, no, let me talk about the pink circles had the sort of total number of reported cases of Lyme disease. And so the biggest number is in Ontario, followed by Nova Scotia. But then the coloration, the darker the green, the higher the rate of Lyme disease per population. So although we have fewer cases than Ontario, we also have way fewer people, and so the proportion of people that have Lyme disease here is high. So this is important for Nova Scotia, right? Understanding the ticks that can cause Lyme disease or transmit Lyme disease. And unfortunately for us, it's getting more dangerous, right? This is a map from 2017 from the province showing estimated risk areas for Lyme disease, and the darker blue are the higher risk. And there's places where the ticks have been a problem for a long time, like the South Shore, right? And then you come to the up-to-date map, and it's just every county in the province is high risk because the ticks are moving further north. So ticks are becoming more prevalent across the province, and that's very likely associated with the changing climate. Um, what causes Lyme disease? So it's not the ticks themselves, but the ticks carry this bacteria called Borrelia burgdorferi, which is a little bit of a mouthful, but it's a cool looking bacteria. This bacteria, so here's a really zoomed in version of it on the left. And it actually has like a spiral cell structure. So this is a single cell and it's in this spiral. And it's tiny. So the scale here, that's 1 micrometer. So if you think again about that millimeter on your ruler, it's 1,000 times smaller than that. So we can't, these images are taken using microscopes. We can't see them with our regular eyes, even if you have fashionable glasses like I do. And then the image on the right is showing um, a, a regular, what do I want to say? It's a cell, it's an animal cell, so similar size to the cells in your body. And then on the outside are all these little spirally Borrelia bacteria. So that's just to give you a sense for the scale of what causes these problems. So Lyme disease occurs when this bacterium makes its way into a human body and starts going through its life cycle and getting into different places within the body. Okay. Now, not all ticks will carry this bacteria. So here are sort of three of the species you can find in Nova Scotia. And Nova Scotia has a pretty good tick safety website, which is where this image is from. Um, the and I've seen the one on the left and the one on the right. I've actually never seen the one in the middle, but it does exist here. So on the left is a black-legged tick. This is specifically a female. And they're kind of very striking. They have sort of this brown, this like orangish abdomen and a dark head region. And this is the species that carries Borrelia, which can cause Lyme disease. Um, they're also called deer ticks. I will call them black-legged ticks because the common name for this critter on the right is dog tick, and I just get dog tick and deer tick confused. So we'll call them black-legged ticks for this talk. But if you hear people talking about deer ticks, it's the same species, and the scientific name is Ixodes scapularis, so which is a pretty cool sounding name. Um, in the middle, we've got a groundhog tick, and which I've never personally encountered, and then the dog ticks, which are what I've mostly encountered. I've seen way more of these guys than these guys, and that's partly because these are what you tend to encounter the most right in the summer when people are out hiking more and doing things like that. Whereas the black-legged ticks you will find more, they're more abundant in the spring and fall. You can encounter them year-round, but just sort of probability-wise, you're more likely to encounter a dog tick. And those of you who have dogs and walk them around, I'm sure know all about this. Okay. Oh, and there's a dime for scale. So these are pretty tiny. Yeah. Um, on the This is just another view of the black-legged ticks. So there's our female again. The males look a little less dramatic. They're just sort of mostly a dark color. And then on the very left here are the younger stages of the ticks. So the nymph, which is the stage right before they become adults, and the larva, which is even earlier than the nymph. And they are this is kind of to scale, so the nymphs are smaller than the adults. And I did, yeah, my cat is not an out, my cats are not outdoor cats, but we take them out on leash around the yard when it's nice. And yeah, one of my cats got bitten by a black-legged tick nymph in my yard for the first time this summer. So that was surprising that they're here. Um, and how do we interact with these ticks? So I've got up top that the, black-legged tick season is spring and fall. Like I said, that's when they're most abundant in the environment. But you can encounter them in the summer and you can encounter them in the winter. Unfortunately for us, like a lot of insects in winter will go dormant, like the way the bears hibernate. They will just like hunker down and not move and just wait out the winter. If it gets warm enough for these ticks to move, they will just start moving and try and find a nice animal to latch onto and drink their blood. So I think the temperature is sort of threshold is 4 or 5 degrees. So if you're out on warm winter days, you also gotta kind of be aware. But when they're ready to when ticks are trying to find an animal to hang on to, they do this what's called questing. That's the image on the left. So this is a tick on top of a um, some vegetation, and they kind of hold their hands up there. If you've been sitting on the couch, you need a bit of a stretch, you can get up and do some questing. Um, but yeah, this is on the right is a slightly complicated version of the life cycle, but it's kind of um, I just wanted to mention it briefly because I think it's actually cool. It actually takes them 2 full years to complete their life cycle. A lot of insects take 1 or less than 1 year. But the eggs hatch in the spring of year one, and the larva, in order to like transition between life stages, so we've got larva, nymph, and adults, they need a blood meal. So the larva will hop onto a warm-blooded host, like a deer or a bird or a deer mouse. Um, and this is where they can potentially pick up that bacteria that causes Lyme disease. So if the animal it bites into has the bacterium, then it will probably get into the tick's system, right? And then the larva um, will grow up into a nymph, and then the nymph um, will also need to feed on a host, uh, a warm-blooded animal to make its way to the adult stage. And then the adults need to feed on a host um, when in order to complete their reproduction. So an adult female, in order to lay the eggs, needs like a nice blood meal. So they're little vampires, these guys. They need a lot of blood. And they do when if you I'm sure many people have seen a tick that's actually had a blood meal. They get huge, um, which is yeah, just amazing biology. But at any of these stages, right, where a tick is biting an animal or a human, that can result in the tick picking up the Lyme disease bacteria. And then if that tick bites a human, then that will transfer the bacterium into the human. So the nymphs are often fairly active in the spring, and the adults are fairly active in the fall, in the second year of their life cycle. There was something else I wanted to say about these guys, but it has escaped me. So, da, da. Oh yeah, I mean, lucky for us, for whatever reason, the ticks, don't bite right away. Like if you find a tick on yourself and it's still moving, then it hasn't bitten you yet. So usually you've got a fair bit of time to like find ticks before they bite you. So that's a good thing to help with staying calm in those moments. Okay. Okay. And sort of my last sort of general tick thing I just wanted to mention before I get into the science that my lab is doing. There's this wonderful website called etick.ca. They've got a whole lot of resources about ticks and how to identify them, figure out what species you're looking at. And they have a place there to report tick sightings. So if you like contributing to scientific efforts, um, they make it pretty easy, and you can actually report on like which ticks are in your area and when, and help keep other people safe. And this map is showing some abundance numbers for Ixodes scapularis, the black-legged tick. So they've got an interactive map. You can filter by species. It's a pretty cool. And there are different ticks in different parts of the country as well. So out here, Ixodes scapularis is the one we need to worry about for Lyme disease, and out west it's something else, Ixodes pacificus or something like that. Okay, but not so many ticks out west. So if you're thinking about moving because of the ticks, that's where I'd go. Okay. So, what got me started on ticks? A lot of my training like, during my PhD and whatnot was working with insects and especially insects that survive freezing. And one of the people who did their PhD in the same lab as me is Laura Ferguson. Great person. She's a professor at Acadia University, and she was doing some work in her postdoc that showed this sort of unfortunate uh, fact that the black-legged ticks, not all of them are carrying Lyme disease, right? Like some of them will have the bacterium in them because they've had a blood meal from a host with the bacterium, and others will not. So in a population, you'll have some with the bacterium, with Borrelia, some without. And Laura and her colleagues found that the ones carrying the Lyme disease bacteria were better at surviving winter. And so that's like a oof moment. That's not great news. Uh, but Laura and I were looking at this and we were like, well, why or how? How do they do this? Right? Because if we can understand how, maybe we can do something about it. So, um, what what we're working on at the moment is this question of like, why are the black-legged ticks that carry Lyme disease good at surviving winter? And this is the team that's been working on this question. So there's Laura and her. I don't know if her kids helped, but sometimes they probably do. And she's at Acadia, and she's had a master student, Mia, who's done a lot of great work on this project. And then over at St. FX, there's me with shorter hair. And two undergraduate students, Amber Goff and Miranda Van Orscott. And each of these students like has done a bunch of the work that I'm going to show you later, so I want to make sure that I acknowledge them. And they're all going on to continue in science, which is great. And I also want to thank the funders real quick. So Laura and I are both funded by NSERC, which is a federal funding agency that we really appreciate. There's also CanLyme, the Canadian Lyme Disease Foundation, that gave us some of the money for this work. And both Mia and Amber were funded. Their like salary was funded through the Scotia Scholars Program through Research Nova Scotia, which unfortunately has just been that program has just been cut. Thanks, Tim Houston. But anyway, I'm glad that it was able to support some great young scientists to do some good work. Um, so let me tell you about what they did. So when we think about, okay, we have some ticks that are pretty good at surviving winter. What do they have to deal with during winter? And here I've got just an image of a different bug, a swallowtail butterfly, because they're much cuter than ticks, showing that winter can be quite long, especially if you consider like fall and spring are quite cold for an organism whose body temperature matches the environment. So winter is long, winter is cold. That's a big challenge. Um, Winter can be very dry. So dehydration is a challenge, and there's less available water because a lot of it's frozen in snow and ice. There can be different, from the perspective of the insect, like they may become more vulnerable to pathogens that can kill them. Like there's different diseases that are more prominent in the summer versus the winter. Um, and it's difficult to find food, which is why a tick might risk like moving around and trying to find someone to bite in the middle of January. And so there's very little food, and starvation is a real risk during winter. So the work I'm going to talk about here is focusing only on one of these, which is cold, and perhaps the most intuitive stress associated with winter. And what we did is we collected ticks from a few locations in southern Nova Scotia. So they were field collected ticks. So we didn't try to rear them in a lab facility. And the collections were in the spring and fall when they're most abundant, partly because they're most abundant, but also to allow us to look at ticks kind of before winter. Are they preparing for winter in the fall? And then spring after winter when they're perhaps at their most vulnerable after having experienced all those challenges. And of these naturally collected ticks, some will have this Borrelia bacterium that causes Lyme disease, and some won't, right? So there's kind of a natural um, experiment happening there. We measured some elements of their cold tolerance, so how well do they survive low temperatures? Then we cut them up, which is, I know how some people will deal with ticks they find on themselves. It's a decent way to kill them. Um, but we cut them up for science purposes, not to murder them, but because we needed to, well, we wanted to look at whether there were any sort of special chemicals or molecules they were accumulating to help survive winter. And we also, in order to figure out, are they carrying the bacterium that causes Lyme disease, we have to do a little bit of genetic work. So we cut the ticks in half. We used half to figure out were they carrying the Lyme disease, and the other half to figure out something about their overwintering biochemistry. Okay, so just to show you what tick collection looks like, this is Amber in the field, pants tucked into the socks, important safety element. And normally when you're out walking in the woods, you do not want to pick up ticks, but if you do, you can drag a light colored sheet with you and the ticks will grab onto it as they're questing. And then you can use tweezers to pick them off the sheet and put them in a tube to bring back to the lab to do those experiments I was talking about. Um, so Amber went out with Mia and some other folks to collect ticks, mostly in the valley, so and sort of the Wolfville, Canterville area, as well as in the South Shore. And we could only do the genetic testing at the very end, right? After we'd done all these other experiments, because once you cut open a tick and grind it up, you can't you can't figure out like does it survive freezing. Um, but I'm going to show you the genetic results first, just to give you a sense for what we're working with. And to do that genetics, each half tick would go into a tube where it would be ground up and have various chemicals added to extract the DNA, the genetic material. And that DNA contains the DNA from the tick, right? But also any bacteria that are inside the tick or in the tick gut or on the tick. So each of these different colors of DNA can represent a different species. And we want to know, is Borrelia there, the bacteria that causes Lyme disease? So we use a procedure called PCR to specifically make many, many copies of the Borrelia DNA. So you can use like the original DNA as a template and make extra copies of it if it's present, So if the Borrelia DNA is there, then we'll get lots of copies of it by using PCR. And then we can actually see that if we run that DNA on a gel, kind of like you would see in CSI. So this is showing sort of two samples here, and a marker here that just tells us this kind of the size of the pieces of DNA. So each of that is a piece of DNA from something we've purchased. And then if there's lots of copies of Borrelia DNA from the PCR, we'll see it on this DNA gel. And if there's no Borrelia present, then there won't be a band. So we can figure out, who's there. And the news is not great. So here on this graph, we've got along the bottom, the x-axis, the different collections. So spring 2023, fall 2023, spring 2024, fall 2024. And on the y-axis, we've got what percentage of the ticks were carrying Borrelia burgdorferi. And I put a dashed line in there to show 50%. And all of those numbers are above 50%. So also we worked with like hundreds of ticks. Those are like how many were in each collection. So that's, you know, at least half of the ticks we collected were carrying Lyme disease, just over half in both years. So that's a little bit scary, but good for our experiment because it meant we would have like enough ticks that had the Lyme disease and didn't have the Lyme disease. So how do they deal with the cold? One way that we can look at how they deal with the cold is to cool them down in a controlled way in the lab. So on the left is it looks a little messy. Sometimes the lab bench looks a little messy, but what we have here is a recirculating chiller, so it can cool liquid in there down to -20, and in a very controlled way. You can tell it like cool at half a degree per minute. It's very sort of specialized for the work that we do in our research group. And this cold liquid cools this unit up top, and then we can put ticks into that unit. And all these little wires here, each one is measuring the exact temperature of each tick. So there's sort of one wire per tick. And what we can do when we measure that temperature is we can get data that kind of looks like this. So here we're looking at temperature of an insect or a tick on the y-axis over time as we control them down, cool them down in a controlled way. And something you might not know about insects and their friends is um, when you cool them down, we know that like water is supposed to freeze at 0 degrees, right? In reality, sometimes it freezes it at temperatures a little bit lower than that. But if you cool down an insect, it's probably freezing at -4 or lower, depending on the species. It could be -4, it could be -20, anything in between, and sometimes even lower than that. So at some temperature below 0, this organism is going to freeze, and we can tell that it freezes because when ice forms, it releases a bit of heat. So if you're tracking the temperature of this insect, it's dropping because the environment is getting cooler, the insect is getting cooler, and then ice starts to form, a little bit of heat released, and then it will continue to cool. So the temperature at which ice forms is called the supercooling point for historical reasons. It's a bit of a strange name. You can think of that as like the freezing point. One thing we can do is measure, if we take these ticks and cool them down, what temperature do they freeze at? And we can use that to help understand how they might survive low temperatures in the wild. Okay. And this is what the data looks like. So we've divided it up into females on the left, males on the right, and I've separated out fall and spring for each sex. And on the y-axis, we've got the supercooling point, the temperature at which they freeze. And each dot here is one tick. So you can see kind of all the ticks there, and the boxes just kind of try to summarize where most of the ticks are. So within the box, you have sort of at 50% of the ticks are in that box. And one thing that is really striking about the ticks is that this is a huge range. So we have a tick here that's freezing at -2. We have a tick here that's freezing at -17, which it's a bit strange. Like in a lot of insect species, you'll have a more narrow range. It'll be like maybe everyone freezes between -10 and -15. So it's kind of crazy to have such a 15-degree difference. But one of the possible reasons for that is, these ticks were collected from the wild, and we don't know their age. We don't know when they last ate. We don't know when they last mated. There's lots of things that could maybe affect their biology that could then affect the temperature at which they freeze. But the thing that we care about overall, ultimately is like, well, what about the ones that are carrying Lyme disease versus the ones that aren't, right? So the color coding of these points, the filled triangles are ones that are infected. They're carrying Borrelia burgdorferi and they can give you Lyme disease. And then the open circles are uninfected. So they don't carry the Lyme disease. If they bit you, it would suck, but you wouldn't get sick. So uh, what you can see is like those circles and triangles are kind of evenly spread out for and whatever place in the graph you look. And when you run the statistics, there's no difference between infected and uninfected ticks for the temperature that they freeze at. So this is not going to tell us why infected ticks survive winter better. Sometimes that happens in science. We definitely expected to see a difference, and we didn't find one, but we froze a lot of ticks, so we're confident in our data. And no, they seem to be doing the same sort of thing. Okay. Um, but surviving winter is not about, well, what temperature do you freeze at? It's also about, do you survive freezing? Because if you survive freezing, freezing isn't such a big deal. So it's not a big deal if the environment gets colder than your supercooling point. But if you don't survive freezing, then and the environment gets colder than your supercooling point, then that is a problem because you will die if you are the tick or insect in question. So what category do the ticks fall into? The way we figure this out, we use the same instrument I talked about before, and we cool a bunch of ticks down all at once. And we cool them down to a temperature at which half of them have frozen. So half of them hit their supercooling point and freeze, and the other half have not. So if we look at the graph here, if we look at this box here, if we cooled them down to, let's say, -10, half of them would have frozen because their supercooling point is above -10, and half of them would be unfrozen. And then we can see, okay, they've all been cooled to the exact same temperature, exact same conditions, except half of them experienced ice in their bodies. And then you can tell, um, is it the freezing that kills them or the temperature? So what the data looks like here, I'm just showing you females, but the males had a similar trend. And we've got the spring data on the left from 2023 and 2024, and the fall data on the right. And each of these bars, gives you the proportion of ticks that froze or didn't freeze and survived. So for example, if you just look at spring 2023, a bunch of ticks froze, but 0 of them survived. You can see that line there. And the ticks that, half of them didn't freeze, and about a quarter of them survived. Okay. So, and we see something similar in spring 2023 and spring 2024. And the temperature here is the temperature they were cooled to. So we had to cool them down to about -10 in order to get half of them to freeze and half of them to not be frozen. So that's not great odds for the ticks. Basically, they're dying. Most of them are dying, whether they freeze or not in the spring. Right, all this white space here is indicating dead ticks. But in the fall, things look up for them. So the frozen ticks still die, but the unfrozen ones, most of them survive. And the survival was a little bit better in 2023, where we cooled them down to -12.8. In the 2024, we had to cool them down a little more to get half of them to freeze, and a few more of them died. But this suggests that in the fall, when these ticks are preparing for winter, they get quite good at surviving low temperatures as long as they don't freeze. So they don't survive freezing, but if you keep them above these temperatures like -12, they're probably going to be okay, at least for a short while. And in the spring, we would call these guys chill susceptible. So very few of them survive low temperatures. And that's quite possibly because they've been through a long winter and they're kind of on their last legs. Okay. Um, but Jantina, you might be saying, what about the infected versus uninfected ticks? They're not even differentiated in these graphs because, again, there was no difference between infected and uninfected ticks. So, um, I'm going to talk a little bit. Okay, so maybe at the whole animal level, their ability to survive low temperatures um, just isn't that dependent on whether they're carrying the Lyme disease bacterium. Are they doing anything inside their bodies that's making them survive better? And here, I am just keeping an eye on the time. I think I have time to talk about this. One thing that lots of insects do to prepare for winter is they accumulate these chemicals called cryoprotectants. So this is a goldenrod gallfly that is very cool freeze tolerant bug. And this is the chemical, they accumulate these chemicals inside their body, in their blood, um, during the fall. So here we have September, October, November, December. We're going through the year here. And then on the wax, this is just the concentration in their bodies. And you can see we've got this increase in both of these molecules. And this coincides with, at the beginning of fall, they don't survive freezing. But by the time winter really gets going, they are freeze tolerant. They can survive freezing. And these molecules are helping them do that. So and glycerol is this is what it looks like for anyone who likes looking at the chemical structures. Um, it's about the size of, it's like half of a glucose molecule. And it's really good at helping lots of organisms survive winter. So we looked for it in the ticks, and I'm going to skip the methods slide here, but if anyone really wants to chat about it, we can. We looked for glycerol. We also looked for some other common cryoprotectants in the half ticks that we had dissected. And the only one that really popped up was glycerol. So the other ones were at very low concentrations. And here again, we have females on the left, males on the right. We're looking at fall and spring in both cases. And on the x-axis, we've got the concentration of glycerol. And it was really low in the spring in both males and females, and much higher in the fall in both females and males. So this is likely something that they are accumulating in preparation for winter to help them survive winter. Now, we haven't tested that glycerol is required for them to survive winter, but it's certainly correlated with it. So it's a good sort of hypothesis. But once again, there was no difference between the ticks that were infected and the ticks that were uninfected with Borrelia, the Lyme disease bacterium. So this was like we learned a lot about the ticks, right? We learned that as they get ready for winter in the fall, their ability to survive low temperatures improves, and they are accumulating molecules like glycerol to help that. We also learned that a good half of ticks in southern Nova Scotia, at least, the black-legged ticks are carrying Lyme disease. But we did not learn why the ones that carry Lyme disease seem to be better at surviving winter. So that just means there's more to do, which is kind of how science works, We have a hypothesis and we don't find support for it. We have to generate new hypotheses and try new things. So, we only really looked at cold and dealing with cold, but maybe the Lyme disease bacterium helps with dealing with some of these other challenges associated with winter. There's also the chance that the Lyme disease bacterium doesn't help the tick at all, but it's just correlated with something. Like, maybe the ticks that have Lyme disease are all feeding on a particular species. Like maybe they're all feeding on deer mice and they get the Lyme disease from deer mice. And there's something about deer mice blood that helps them survive winter, right? So it could just be that the two things are correlated. And it would be, yeah, at some point, I hope we figure out what it is. But for today, I don't have an answer for you. So yeah, I think that leaves us a little bit of time for questions if there are. And thank you very much for your attention. And stay safe out there.
All right. So first and foremost, I want to ask everyone to give a nice virtual thank you to Jantina for that amazing talk. It was very, very informative, and I definitely have a lot of questions for her, but I will hold them and I'll ask that for those of you online who do have questions, can you just please use the options under React to raise your hands, and we'll kind of go based on that. And so let's see. The first one I see here is Helga. Please feel free to unmute yourself and ask your question.
And I'll lower my hand. That was lovely, Jantina. I really enjoyed that. My question is, you might have said it, but I didn't really catch it completely. How big was the difference in survivorship that Laura found in wild ticks, whether or not they had the Borrelia? Because it depends. If it's a really big effect, you would expect to see something in the mechanisms you use. But if it was like a 5% difference, or I didn't catch how big the survivorship difference was.
That's a great question, Helga. So that original study that kind of inspired this, I don't have the number off the top of my head. Um, so that's something I would have to look up. But you're right that it could be if the isn't huge, then it might be quite difficult to figure out why.
Mhm.
Yeah.
Thank you.
All right. I think Carol Morrison was next. Apologies to Jen Priddle if I'm getting the order wrong.
Uh, okay, I wondered if you were familiar with the work of Dr. Paul Manning at the Truro campus of Dalhousie University. He's sort of an insect guy, gave a presentation to our garden club, and he has a graduate student, Alex Indigo, um, who's got all of these garden clubs, got all of us to collect the damn things, put them in our freezers, and they're looking for possible um, things that are going to counteract the ticks.
Wonderful. Okay. I do know Paul. He's a great guy. Um, and very engaged in like, yeah, I like how you said that phrase that convinced you all to catch the darn things. Um, and yeah, if
So
we're
what's
trying to understand like how to deal with the ticks long term, yeah, it would be great if there was a natural predator that we could encourage to eat them. And yeah, I'll keep an eye on that. That sounds really cool.
apparently we're not we're not allowed to do it, but a couple of possible predators have come over from the States, and they're in New Brunswick, and sort of hopefully they're coming up here. So yes, we would love to, because they've been absolutely devastating to us gardeners in this area, at least. I'm in Oakfield, near the airport.
Yes,
So
I miss the days when I could go out in the garden and not worry about ticks. Yeah.
definitely.
Yeah.
Okay, just a quick comment before Jen answers questions. For those who are willing to, guinea fowl are very good. They are very loud, but they are a good option to deal with some ticks. Anyways, Jen Priddle, please.
Hello, doctor. It's actually Greg here. Wanted to ask a question. Curious as to whether or not the fact that we've had a fairly severe and early starting winter with a number of days where it has been, well below -15 or -18, does that give any hope for some devastation to aforementioned tick population in the coming season? Thank
That's
you.
a that's a great question, Greg. Um, yeah, the low temperatures may help, but it depends on where the ticks are. Because if they're under snow and fairly protected from that, then many of them will survive.
Boo.
But some of them will freeze and die. Um, but probably not all of them.
Hey, Shelly Adamo?
Oh, perfect. Yes, so thank you. I really enjoyed that. I don't know if you guys can see me, but that's okay. The question I had, Janita, I don't know if you're thinking of doing this already. I mean, you were looking at the very standard, reasonable compounds to look at for surviving the winter if you're an arthropod. But there was that paper by Neela Kanta, and I know you've talked to Laura about this, I'm sure, because I'm not sure she completely buys it. But wouldn't it, are you thinking that maybe you need to look at the proteins, not, the metabolites, basically? So, you could do fairly easily, I would think. We have the service down here at Dow, as a matter of fact. If you wanted to look at either take the blood from the ticks, there's different ways you can do that. And actually look at the protein, the proteomics of it, and see whether you get the same thing. She found, as you know, Neela Kanta and their team found that one of the things that's different about infected ticks, it wasn't Borrelia, it was a different pathogen. It causes an increase in this molecule that's like a molecular moonlighter that can be either a cryoprotectant or an antimicrobial protein. And so it's basically by increasing the production of that, it's sort of just a byproduct, ends up having a greater success. And just a question for the other question about, because it was from my lab actually, that was the study that looked at the overwintering and the increase in female survival. And it it was more of a correlation because we didn't infect the females. We just found that for a number of potential reasons, the females that were infected were more likely to survive, and it was a fairly large effect. They were like 30% more likely to get through the winter alive. It varied a bit depending on the year.
Okay, yeah, that's awesome. Thank you, Shelley. I should have punted that question to you, but that's great. So that answers Helga's question about the effect size of that. And yeah, that there may, in terms of like how insects and other related organisms survive winter. Yes, it's certainly not just about those small molecules like glycerol. So there is this potential antifreeze protein or antifreeze glycoprotein that's been detected in these ticks. And yes, I am similarly skeptical about the original paper that you referenced, but it's worth looking for in general to try and understand some of the other possible ways, because there's lots of like little mechanisms that can contribute to better cold tolerance. So we certainly didn't hit all of them in this first suite.
No, I was just going to say, because I don't know if SaneFX has its own proteomics or whether you can do proteomics in-house, but Dalhousie's system is up and running, so it's not even very far away. So I just mention it.
Okay, awesome. Thank you.
Okay, I think Helga has another question. Oh, Helga, you're on mute.
Okay, do you hear us?
Yep, you're good.
Okay, where do the ticks, do they have a behavioral pattern? Do they dig into the leaf litter in the winter, or they always stay where they might have contact with warm-blooded organisms up on the surface?
That's a good question. They're generally close to the ground during the winter, but if it warms up, they will climb up on structures that they can use to latch onto a hiker's leg.
Yeah.
And did they have any particular preference for type of vegetation? Are grasses more likely or than fir boughs and things like that, or anything like that, or is that known?
I've encountered them most in grassy areas. But I don't know if Shelley, you wanted to weigh in on that.
Yes, well, it's funny, it's a bit controversial, but deciduous tree leaf litter seems to be one of their faves. So maple oak, for example, they really like that. Grasses, yes, but for overwintering, they really do seem to prefer that leaf litter. What I can tell you is they don't like balsam fir needles, so they don't like to overwinter under those. But they will over they do quite nicely in pine, for example. So there has been a fair bit of work actually looking to see whether there's a good relationship between overwintering survival and substrate. And basically they need to be under something. They need to be under leaf litter or they need to be under turf. And they do even better if they can get some nice snowpack that Jantina already talked about. But there's been some nice work out of Maine just on looking at how important that is. The other thing, though, that I find really interesting is Burtis's work, too, that showed that you think that starvation would be an issue for them and that they'd want to come out and look for food. And that's what's pushing them to come out and quest in really suboptimal conditions in winter when you think about it. But actually, interestingly enough, he found that they hardly lose any lipid reserves at all for the 3 months of winter. This is in upstate New York. So they seem to be really good at conserving their lipids over the winter, assuming, I guess, whether or not that they quest more. And I don't know if that's another area of research you might be interested in, because I think it's one of those great trade-offs. If you look for food a lot, then you probably would run down your reserves faster. But you also may be looking for a mate. You may be looking for food. Does it matter how much reserves you have, what strategy you pick? I don't know. I just, I'm not doing things on overwintering ticks anymore, but these are one of the many questions I hope you get a chance to tackle.
Can I just interject one more question? It seems
Sorry, Helga. Alex F does have their hand up. I just want to be mindful of time, and I want Alex to have the opportunity to answer the question.
I'll put my hand up again.
Thank you.
All right, well, this will be hopefully a quick one, a simple one, hopefully. So you froze, if I remember correctly, your ticks to like around -12 to -14 Celsius. I wonder if that's too cold, since Wikipedia tells me the average temperature in Halifax is between -0 and 8. So I wonder if you'd freeze them at a lower temperature, whether you'll start seeing a difference emerging then. Like -14 is too cold for even the Lyme disease power ticks to survive.
Yeah, okay. That's a great question. Thank you, Alex. So yeah, the way that we kind of measured survival after freezing was, yeah, a little bit artificial. Like we're cooling them down in the lab faster than they would cool naturally as well. Um, but yeah, we've also got some data looking at more realistic exposures, like exposing them to -5 or -10 for a few hours. And they if you put them like straight into those temperatures, like if you just take them from 4 degrees where we're keeping them and like pop them straight into -10, they don't do very well. But if you cool them down slowly to -10 or -5, um, like they would experience in a natural environment, they can survive those temperatures just fine. We don't have the data on how infection interacts with that. But that would also be a next step, would be looking at some of those more ecologically relevant exposures.
So a regular tick can survive -8. That's not very comforting, but thanks.
Sorry. And all of these are still short term. Like if I don't know how they're doing if you keep them at -5 for 4 months. Like that's a different story, right? And if we had more ticks, we would have loved to do some longer exposures. So we'll have to do that in a future year.
Great, thank you.
All right, Helga, please ask away.
Okay. It seemed to me like I've been in Nova Scotia now for 15 years, and it seemed to me that when I was first here, ticks were really abundant, and they hadn't moved north yet. But they seem to have moved north, and I kind of feel like they might have left us behind. That things are kind of like I haven't seen a tick, and I'm outside all the time. I haven't seen a tick for a while. Like maybe I saw one or two.
It mainly has fir trees, I think.
Yeah, so I just wondered if there's something like an invasion front that as the ticks are marching north, that they're really, using this new habitat and filling it up. I mean, it's just a crazy idea, but it goes with my observations that there were lots in Kingsburg 25 years ago. And then there were quite a few in this area as well 15 years ago, but now there seem to be fewer. But
that's also a great question. I don't know.
I don't know.
I hope, well, and it's, from a public health perspective, I think we're still worried across the province, but it can certainly vary year to year.
Yeah.
And I don't know how some of the things like the fires and the droughts
Oh
and
my
things have
God, yeah.
affected the ticks.
So many aspects. But I think this is really cool work that you guys are doing. I'm happy to see it. Thank you so much. I will stop asking questions.
Thank you for your questions, Helga.
All right, so just for time, I think we should be kind of winding down. But Corey, I do see you there, so I'd like to give Corey the opportunity to have the last question.
My question is more about my dogs. I get flea and tick medicine from the vet, and it's quite expensive. And how come they have flea and tick medicine for dogs, but not for humans? Or do they?
Yeah, that's a great question. And no, so far there isn't one that's approved for human use, I don't think. Um, I don't know if anyone's, yeah, I'm not sure. I'm not so tuned into the sort of the human health side of it. But so far, like Lyme disease is just treated after you've been bitten. I hope someone's working on it. Because we're not that different from dogs and cats. Like it's you know, when you come down to the basic biology. So yeah, I hope someone's working on it, but that's all I can say. Yeah.
Thank you.
I might be able to answer Corey's question, and there are some clinical and preclinical trials using mRNA, the same technology as the COVID vaccines for Lyme disease, but there has been a lot of mixed results currently. So when it may come to mass production, I have no clue. All right. So, with that said, again, cognizant of time, and I know some people may have a lot of questions. I will be emailing you some questions, yeah, and Tina later on. But again, for those of you who are still here, I'd like to again ask everyone to give a very warm thank you, a virtual thank you to Jantina for this amazing talk. And for those of you who want to relive this talk, again, just like with all NSIS talks. We will be posting it later on to our YouTube channel. And apologies for the background noise of two stir crazy children who have not been outside enough apparently. And I will officially call this an end to our NSIS talk. Thank you, everyone.
Thanks, everyone.